I don't, for what it's worth.
Is it amino acid?
Interestingly,
Right. Many of these things have.
So it was very beneficial for...
My name is Matt Caberlin, and welcome to the OptiSpan podcast. Hey everyone, welcome to the OptiSpan podcast. As you can see, I am joined today by my arch nemesis, Dr. Charlie Brenner, for a no-holds-barred smackdown version of
When
the
did
podcast.
we become nemesis?
No, it's seriously, I'm really grateful that you came out to OptiSpan HQ, came all the way up to Seattle today for the podcast. I think, some of the people in the Twitterverse or Xverse or whatever it's called now probably think that, that we just argue with each other all the time and we don't agree about anything. And I would guess we probably agree about 95, 98% of the stuff. And then the 2% that we maybe disagree on, we're just both very passionate about and get into debates online.
And maybe we'll learn something from each other
I
now.
sure hope so. Yep. So again, thank you so much for coming. And I think this is going to be a fun conversation. We've got several things we can talk about, and we'll just kind of see where it goes.
Okay, let's get into it.
Awesome. So I think certainly we should talk about mTOR and NAD and then the longevity space in general.
Okay.
Do you have a preference where we start?
We can start with mTOR. We can start with geroscience. You want to start with geroscience?
Sure.
Right? So I'm prepared to say that, so the geroscience, as I understand it, as you know, I come from a background of basically being an enzymologist, a geneticist, an X-ray crystallographer, trying to figure out how NAD works, trying to figure out how, DNA methylation works. And I didn't exactly come from needing to work on aging my whole career. I kind of collided with it, almost non-consensually, one might add. But geroscience, the idea of geroscience is that that the fundamental biology of aging is so important and underlying the incidence and the severity of so many different diseases that you ought to try to target the aging process. And I'd be willing to say that in model organisms, the premise of geroscience is strongly supported by evidence, right? So like rapamycin extends lifespan in multiple model organisms, clearly in mice. So, I mean, to me, that itself and the idea that, targeting pituitary pathways can slow aging in so many animals, that to me says that the premise of geroscience is evidence-based. Whether it's going to be actionable in humans, I don't know. But I'm willing to start there and say I'm not a doubter of the geroscience hypothesis.
Sure.
I don't know to what degree it's actionable for people right now.
Yeah, I think that's fair, given that we haven't proven that we can substantially modulate the biology of aging, at least through pharmacological intervention. So
Right.
I guess I would maybe reframe the sort of geroscience concept a little bit differently, which is the way I think about it is first and foremost, that there is a biological underpinning to the aging process, right? That if you look across the animal kingdom, there are biological mechanisms at play. I do think we may get into this. I think it's an area of confusion whether or not, and I think we probably both agree not, evolution has acted specifically to modulate the rate of aging per se, as opposed to other forces like reproductive success.
Right.
But there is this biology that underlies the aging process, and it is under both genetic and environmental control.
Yeah.
And geroscience, I think, that sort of at least the initial definition is the mechanisms that connect that biology of aging with age-related functional declines and disease. And then the hypothesis, to the extent that it's not really a hypothesis, but the geroscience hypothesis is that if you can target the biology of aging, you can have an impact on multiple age-related diseases and functional declines simply by targeting the biology of aging. And as you say model organisms, I would just say animals, right? If you look in all of the animals where this, and single-celled organisms, yeast, and we'll probably talk about yeast, but in all of the organisms where this has been experimentally tested, we can say with certainty that we have identified genes that modulate apparently the rate of aging and certainly lifespan, and that when we modulate those genes to increase lifespan and seemingly slow biological aging, most, if not all, of the functional declines and diseases that go along with aging are delayed or prevented in those animals.
But it's not a wonderful thing to be a dwarf mouse, right?
Well, not in the wild. And you're right, depending on the severity of the mutation, even in the laboratory, they have to be co-housed with. Yeah, I think there are selective disadvantages that go along with severe longevity mutations.
So I think, I saw some of your notes and about, are there longevity genes? Yeah, there's even monogenic longevity genes, right? So in mice, if you interfere with a pituitary pathway, either by disrupting a master regulator of pituitary transcription, or with growth hormone, or with growth hormone receptors, you basically can dramatically extend mouse lifespan. You can extend mouse lifespan, you can extend rat lifespan, but those are tiny little animals. As you say, depending upon the severity of the mutation, they can fail to sexually mature, right? They don't have good thermoregulation. The males would be bullied and wouldn't have access to food, so you sort of have to pull them out of the cage and give them some adopted sisters to keep them warm and allow them to have access to food, and then they can live a very long time, right? But so are there longevity genes? Yeah, growth hormone and growth hormone receptor genes are clearly longevity genes. But, you and I wouldn't trade our genetic, architecture for, having two inactivated copies of growth hormone, right? In fact, one of the funny things that you see when you go to certain longevity conferences is that what you and I have been at some, and we'll sit down at a table with someone that calls themselves a longevity doc, right? A practitioner. And a lot of what they do is they give people, steroids and growth hormone, right? Why? Because, and some of my neighbors in Los Angeles, like if they have a film shoot in 6 months and they want to bulk up their muscles, they might be taking more growth hormone. They might be taking more anabolic compounds, right? Which we know genetically is going to accelerate their aging.
Well, I don't know. So again, there's a lot to unpack there. So I do think, and we can come back to this, but I do think it is, so having lower growth hormone signaling through development absolutely slows aging during adulthood. It's not clear that lowering growth hormone signaling in adulthood actually slows aging, or that increasing growth hormone signaling accelerates aging in adulthood. So there is this question of timing that I think is fundamentally important. So it really isn't clear. Like, I don't know of any evidence to support the idea that increasing growth hormone or testosterone, in fact, testosterone is an interesting case where I think that might actually have positive benefits, certainly for health span for some people, but that increasing growth hormone or anabolic signaling in adulthood actually accelerates aging. I don't actually think there's much data to support that.
Well, it's not the most placebo-controlled data, but the experience of a lot of athletes, in the NFL is not very good.
Sure, but that's obviously an artificial situation, right? I mean, where, first of all, you're probably taking very, very high doses of these things and beating the crap out of your body, right? So again, I don't think we can draw any real conclusions from that. I don't know the answer, but I don't know of any convincing data that sort of moderate increase in those pathways signaling is going to accelerate aging. It probably, and it may not actually do anything to the biology of aging. It might increase your risk for certain cancers and decrease your risk for sarcopenia. So it gets complicated. But I do think it's an interesting observation that the very long-lived rodent models of reduced growth hormone signaling clearly are not, that's not a high quality of life in the natural setting. They would not survive, no question about it, and even in the lab. But having said that, you can look at certain animals where there has been enough evolutionary time for that reduced growth signaling.
Talk about dogs, right?
That's a perfect example, right? Where if you start from a wolf and we now have small dogs that seem to age more slowly, but in general, I don't think most people would say that's a terrible quality of life in the small dogs compared to the big dogs. Now, I'm a big
Yeah.
dog person,
Uh-huh.
so I might
Yeah.
like big dogs better.
Yeah.
But so I do think that there are ways to modulate the biology of aging through these growth promoting pathways that don't necessarily, at least over evolutionary timescales, have to come with
Yeah.
those severe
Okay. So this is
trade-offs.
another point of similarity. We both love dogs. And I promised, I think on Twitter, um, 3 reasons why I think dogs are so important for aging
Yeah.
research, right? So one is the kind of survey data that, and you really have been a leader in that, of dogs are genetically heterogeneous, right? There's hundreds of different varieties. They live in thousands of different environments, right? They live with, rich people, poor people, live out on the street, in park-like settings, on farms, in apartment buildings, and they eat all sorts of different things, and they have different sizes and different genetics. And just getting some information about their health span and their lifespan is really important, because they're a complex vertebrate. Um, they share environments with us. In some many cases, they eat some of the same food that we eat,
Right.
scraps from the table, right? And so that's incredibly powerful. And then number two reason is, in part because of this issue that bigger dogs, which have more growth hormone signaling, live shorter lives, which is tough for us both as big dog owners. Um, they're using the
What
geroscience
kind of dog do you have, by
hypothesis.
the way?
Um, she's a mutt. She's a rescue. She's got some boxer in her and a bunch of other
Yeah.
stuff.
Well, that's good from the perspective that mixed breed dogs, based on body size, live about a year longer than purebred
Yeah,
dogs.
but she's
So
11, and there's some lipomas going on, and we had one of them excised, and it was attachment to muscle. So
yeah,
it's a sad
I know.
time
Believe
for
me, I
for
know.
me and my
I get it.
my wife and stepkids, and to see what could be coming our way in the
Yeah,
next couple of years, right?
yeah,
But but there's, as you know, you may even be an advisor of Loyal,
right.
right? That there's drug development, in this space to see if, with a mature, large dog, whether you can dampen growth hormone, signaling, and potentially extend health span,
Right.
right, in dogs. And then the third reason that I think more people should talk about is, what are the longevity interventions that a human being can do in 2024? And one of the best things to do is take your dog for walks, right?
Right.
Because we're not active enough, right? A lot of our dogs are not active enough, and so get that dog on the leash. The dog has you on a leash, and get some treats and enjoy, nature. And so dog walking is probably, if you were to compare people that have animals that need to be walked outside, or you have to run around with kids, and you do a survey of health of people throughout the decades, you would see that people that are more active, that spend more time outside with their dogs and kids, are more healthy.
Yeah, I think you're almost certainly right there. And one of the things that we have thought about a lot at the Dog Aging Project, but haven't really been able to make happen yet, is to pair the Dog Aging Project, which of course is a longitudinal study of aging in dogs, with one of these human longitudinal studies, and get some of the human participants in the human longitudinal study that have dogs to
Yeah.
participate in the Dog Aging Project, kind of start to look at
Yeah.
in a more quantitative way at those kinds
Yeah.
of
Yeah.
questions. But
But
absolutely.
people are hungry for interventions for ourselves
Yeah.
and for our pets,
Yep.
right? And I just get, I don't like the, sort of predatory idea that is sometimes propagated, that we already understand aging, or that we already have human interventions for aging that work, and that you don't have to age, or your dog doesn't
Sure.
have to age, and those things are
Trust me,
just commonly
I agree. I
commonly
push back against that too.
overstated.
Yeah, no, I totally
Commonly
agree.
overstated.
So actually, this raises an interesting question, though, which is, what is your thought about? So does it change your optimism about an intervention if we get to the point where we have an intervention that's shown to convincingly in a clinical trial increase lifespan, improve health span in dogs, does that increase your confidence that it's likely to have the same effect in people as compared to mice, for example?
Oh, greatly. Oh, greatly. I mean, one of the things with mice is that mice are really small and they have a super fast metabolism and they have a very short lifespan, and they can get by in a small cage without needing a huge amount of brain power, right? They don't have to find their own food. They don't have to compete for food. They don't die of falls, right? And so they're a remarkable model because we have such great control over genetics, right? In a mouse, but they're so confined that there's limitations in the model of what you can learn.
So let me ask this a different way. So, is it your opinion that something that, like growth hormone signaling, clearly modulates the biology of aging in mice in a way that seems to slow the aging process, right? I think we both agree on that.
Yeah.
Is it your feeling that same intervention is unlikely to work in people because it doesn't modulate the biology of aging the same way in people, or because it is such a severe negative consequence associated with doing that even if they're aging more slowly, you're not going to have quality of life or longevity?
Well, we should talk about Laron syndrome.
Yeah, right. That's kind of where I'm going.
Yeah. So the thing is, the Ames dwarf mouse, right, appeared in a lab. It was maybe Ames, Iowa, might have been Iowa State, and the Snell mouse appeared somewhere, and mouse fanciers had some of these very small, long-lived mice, and then the geneticists went and figured out what the genes were, and they discovered this whole growth hormone receptor pathway, pituitary signaling. So there's populations around the world where there's really, really small people.
Right.
And one of them is in growth hormone. Right. Is it
so growth hormone
or
releasing hormone receptor
is in a receptor, right? And that's Laron syndrome. And again, it's one of these monogenic, autosomal recessive, diseases where people are really small.
Yep.
Right. And then some aspects of their blood work is remarkable, right? Their cancer incidence is really low.
Yep.
Their level of psychological trauma is very high.
Right.
Their rate of depression and suicide is very high.
Right.
They don't have great endocrinology in general, right? And so some people would say that the growth hormone story doesn't translate because people with Laron syndrome, humans with the same kinds of mutations, aren't super long-lived.
Right.
However, you're asking a nuanced question because we're not going to inactivate growth hormone in dogs, and we're not going to try to inactivate growth hormone in people. We're going to try to modulate it in some way.
Yeah. Not
me, because my lab doesn't do that. But if people try to do that, I mean, if Loyal is trying to do that, or if longevity drug companies are trying to do that for people, they're going to try to modulate it in some way where you still have muscle mass, but in some way you try to dampen down on the negative effects of it. And I don't know whether that can be done. I don't think it will be easy.
So a couple of things to say there.
Yeah.
So just because I know some people will comment on this in the comment section for the sake of being precise. So the Loyal drug is actually targeting IGF-1, which, as you know, is downstream of growth hormone, but just in the case of just to be precise about what's happening. And in dogs, the IGF-1 locus is the strongest genetic determinant of body size. So same pathway, different targets. a couple of things I would say about the Laron data, and this is my reading of the papers that have been published on this, is I actually think you can make a pretty solid case. It's not proven, but a pretty solid case that the little people who have Laron syndrome seem to be aging biologically more slowly, at least in most ways. You can always say in this tissue, maybe not, but much lower risk of cancer, much lower risk of cardiovascular disease. They have obesity, but don't seem to have the metabolic consequences that go along with obesity. I think and then the question is, you mentioned specifically the emotional and behavioral sort of challenges, right, that go along with at least some people who have that disorder. I think the question is that because of the hormonal dysregulation? Certainly could be. Or is it because of the social pressures? And again, this is sort of it's not a uniquely human situation, but certainly in people, body size, we know, stature has this huge social component, right? So it's hard to know if those deficits are more biological or more socially driven. And I don't know the answer to that. But I think at least for some aspects of aging, you can make the case they may be aging biologically more slowly. And if they were in an environment where everybody had Laron syndrome, maybe they would live 30, 40% longer and be healthier longer. I actually think there's a pretty good chance that could be the case, but we're obviously not going to know. So it's just speculation. Like I can say, yep, I think that's what's going to happen. And you can say, no, I don't think that's what's going to happen. I don't think we know. But you're right. I think the strategies that would be taken are adult onset, somehow tweak the pathway. And as far as I know, there's only one study in mice where it was a monoclonal antibody targeting IGF-1, I think, or the IGF-1 receptor. And there was this sexually dimorphic effect where I think it was maybe in females there was an effect and not in males or vice versa. So positive effect on lifespan. So who knows? I do think the one thing I worry a little bit about, and you touched on this, when you get outside of mice, is mice don't seem to be susceptible at least as much to bone density loss, muscle loss, at least in terms of limiting lifespan. In both dogs and people, especially in people, that's a big deal, right? And if you've got a big dog, it's a big deal. It's a big deal in big dogs. And so if it is the case that turning down IGF-1 or growth hormone in adulthood leads to reduced function, muscle mass, muscle function, bone density, there could be side effects that you wouldn't see in mice.
Well, I mean, skeletal muscle mass is really important.
Absolutely.
Right? So, I mean, that's why if you ever take the position, which it sounds like you're considering, that some level of hormone replacement is a positive thing for human aging, I'm sure you'd be taking the position because it would tend to preserve skeletal muscle mass and function, right?
Bone density too.
Yeah. Right. So, I mean, skeletal muscle is a, primary
and metabolic. These things are all tied together, right?
It's the primary, glucose disposal system of the body, right? And it's very important for us to maintain our skeletal muscle mass, which is why we should be out there walking our dogs and doing exercise and, weight-bearing activities, 6-7 days a week, right?
Yeah. And this may be a case where, hormone replacement, and again, that's even a catch-all term because you could mean different hormones, right? But hormone replacement may be a situation where you can, in humans, offset some of the phenotypes of aging, but you're not actually targeting the biology of aging. Like I really don't know how to. I don't know of any data to really think about that one way or the other. I do think I would like to see someone do testosterone intervention study in mice. I've never seen that study done. So as you probably know, in the interventions testing program, 17-alpha estradiol increases lifespan, at least in male mice. And now estriol, so another estrogen, increases lifespan. So that seems to have an impact on the biology of aging in mice in ways we might not expect. I would like to know what testosterone does, because testosterone also goes down in mice, but I don't think anybody's done it. So we'll have to wait and see.
Right.
But I think, again, there's a lot of accumulating data, in my view, at least, in people, certainly on this in women's health, that hormone replacement therapy has a lot of benefits for a lot of women in the context of menopause. I suspect that the same thing is true for a lot of men, but is it actually improving the biological aging process?
Yeah, and it's a nuanced conversation. Neither of us is an MD, right?
True.
But I literally remember where I was. I think it was on the faculty at Dartmouth when some of the first studies that were linking HRT to incidence of cancer, right? And then it just kind of got shut down, right? And these things are, dose dependent and timing dependent, right? And I think a much more nuanced understanding has emerged over the years, and probably too many women shut it down, potentially. And because I think that there are data now that indicate that depending upon, that when it starts, what the route of administration, like topical vaginal estrogen being particularly safe, that it can be really very helpful for a very large percent of women.
Yeah, I would agree with that. And I mean, I think this raises a question that I'd be interested to hear your thoughts on, because I think we both sort of subscribe to the notion that clinical trials are the gold standard, right? We would love to have the randomized, placebo-controlled clinical trial data for everything if we could. But clinical trials can get stuff wrong too. And that can be because of the way the trial was designed, limitations with the trial design, or the way it was interpreted, right? And I think this is a case where you can at least have a discussion around the possibility that the Women's Health Initiative got it wrong when it came to hormone replacement therapy. And so I wonder how you think about that, because I think in the space that, at least in the longevity space, I was going to say the space that we're both in, I don't know if you consider yourself in the longevity space or not, but, there's a lot of back and forth about the idea of, what sorts of data are meaningful and what sorts of data can we use as scientists, or should people use as individuals as they're thinking about ways to improve their health.
Yeah. Well, we know that mom's advice is really good, right? Which means that you should be physically and mentally active, socially engaged, eat in proportion to your size and physical activity, trying to maintain that high physical activity, prioritize sleep, don't do drugs and alcohol, avoid smoking, right? Avoid accidents and violent, situations, right? And that's the gold standard, right? And so overall, drug use is associated with lower lifespan, right? Polypharmacy and having lots of disease and conditions that have to be treated with drugs is associated with lower lifespan. Taking drugs that one is not prescribed is associated with lower lifespan. Not taking drugs that one is prescribed is also associated with lower. So I feel nervous when I see people, taking a whole lot of different drugs and supplements, because generally the cocktail hasn't been evaluated, right? The benefits of particular interventions are usually exaggerated, and the risks of them are usually sort of not, being dealt with. And so that's why, I don't think, other than mom's advice, which I think is really important, and I get back to, dog walking and all that good stuff, I don't think that there's anything right now that you can say is a human longevity drug. Right. And I don't consider NR a longevity drug. It wasn't developed for that purpose. I think rapamycin has a potential, but, I think that there's also, when we talk about skeletal muscle, we got to understand it better, right? Because mTOR, so it's rapamycin inhibits a master regulator of skeletal muscle mass expansion, right? And so, I'd love to know the long-term consequences of taking it, and like how you plan on evaluating that.
Yeah, great. So that let's move into rapamycin mTOR. I think that's a good segue. And again, I think, even though a lot of people watching this might think that we disagree about a bunch of stuff, I don't disagree with anything you said. Like I agree with you. There's nothing that we can point to outside of, exercise, nutrition, sleep, reducing stress, human connection that we can say with confidence is very likely to improve your health span and or lifespan. There are things that you and I probably have higher levels of, if we had to put a probability to it. But I think, if we're being honest, we just don't have data for things like rapamycin or NR or pick your favorite other supplements. So this question of rapamycin and mTOR and skeletal muscle, let's come back to it. I think maybe what I'll do is I'll give you sort of my view of why I think why I'm pretty confident that mTOR and rapamycin modulate the biology of aging, and you can tell me if there's anything you disagree with, and then we can do the same thing with NAD in a few minutes. So, I mean, I look at this from the perspective of looking across the different organisms where mTOR and rapamycin have been studied. And we see that, in yeast, nematode worms, fruit flies, mice, and then now we have, I think, pretty solid data in marmosets, so non-human primates, that you can reduce mTOR signaling and increase lifespan from an interventional perspective.
The marmoset data not being published.
That's right. So this is the data that Adam Salmon presented at the AGE meeting.
Okay.
Right. Yep. Hopefully soon. And at least in the yeast, worms, flies, and mice and rats, you can increase lifespan by modulating mTOR genetically or pharmacologically, right? Which again, I think just adds to the level of confidence. And as you know, reproducibility is a huge factor. This has been done by, more than a dozen labs, right? So there's that independence component. So the lifespan piece, I think, is rock solid. And then in the cases where we can actually look at functional declines and diseases and molecular markers, organ tissue functional markers of aging, in mice primarily, some in rats, we see that these treatments, again, either genetic or pharmacological, generally seem to either delay the declines that go along with aging, or in some cases actually reverse the functional measures that go along with aging. So again, supporting the idea that this isn't just an anti-cancer mechanism or an anti-obesity mechanism. It seems to be fundamentally affecting the biology of aging. So again, I think in the laboratory, the case is pretty solid. Now, does that mean there aren't any negative consequences that might, offset the benefits? No, we don't. I mean, again, might be that we just haven't looked for them or haven't seen them, or if you come out in the real world, you're going to uncover side effects that counteract the benefits.
Well, Blake Rasmussen, right? I mean, so there's a small clinical trial that was published by Blake Rasmussen, who's an outstanding exercise physiologist, and took, I think it was like 13 milligrams. So you may consider that a large dose.
I mean, it's larger than what most people are doing, but yeah.
So he had healthy men do exercise, right? And the positive control for his experiment is that after exercise, he can measure an increase in protein, skeletal muscle protein synthesis, right? And at that dose of rapamycin, he blocked the protein synthesis in the muscle, right? So that concerns me, right? Because you and I are going to the gym and, trying to maintain our skeletal muscle mass. And so my concern is if people, and this is a question for you, my concern is if people are taking rapamycin, what's the dose that they're taking and what's it going to do to their, beneficial effects of exercise, right?
Right. I think it's a good question. I think we don't know the answer. There's a couple things that I think about here. So one is that that's no different than fasting. Fasting would do the same thing. In fact, it'd probably do worse to mTOR signaling and to muscle synthesis. And yet nobody's worried about fasting. So that's one thing I would say. If you fast one day a week, nobody's really worried about that it's gonna prevent you from being able to maintain muscle mass. I'm not saying that it wouldn't have effects on muscle mass. I'm just saying I don't think there's an equivalency of concern here when fasting is probably more severe than rapamycin in this context. The other thing that I would say as well is that at least the way nobody's going to take 13 mgs a day every day. Okay. So at least the way most people are dosing rapamycin, if it's once a week, even if it's the case that it blunts new muscle synthesis once a week, I think it's an open question. Would that have a net detrimental effect, no effect, or positive effect if you were in a regular resistance training program?
Yeah.
I don't know the answer.
Yeah.
I don't know the answer. What I can
Yeah.
say with confidence is I know multiple people, including myself. Now, I don't take 13 mgs a week, but I've taken between 6 and 10 mgs a week, who have been quite successful actually at modulating their body composition in a way that most people would consider positive, meaning a reduction in fat mass and an increase in lean mass while taking rapamycin. Now, you can always say, well, maybe you could have done better if you weren't taking rapamycin. Again, we don't have the clinical trial data, so we don't know.
Yeah.
So all I can say is you
uncomfortable
can,
because it's
I
a
get
drug,
it.
right? It's a drug. You're not a transplant patient,
Yeah.
right? And so it's not been prescribed to you in any normal way, right?
It's been prescribed to me in a normal way just for off-label use.
Yeah. Yeah, so that just makes me
I get it.
nervous.
And
it's like a 50 or 60 page FDA document, right? You can download it, because when we had this interaction 3-4 weeks ago, I wanted to see what is the literature on side effects, right? And it's a lengthy document, right? It says, older people that may have problems with their liver, or it has people that have problems with edema and, immune possibilities, possible, skin cancer, various other things, right?
Yep.
And so this is all, documented, right? And so at a population level, the idea of people just saying, hey, I heard on a podcast that, rapamycin might be a important longevity drug in primates already. It was reported in a meeting, and I'm a primate, so I need to get some. That just makes me uncomfortable.
Yeah, I get it. But I would say there's it seems to me at least, there's a couple of different things going on there. One is your level of comfort because it's a drug. And so my perspective there is I don't really think of pharmaceutical drugs as really all that different from over-the-counter supplements or GRAS compounds. We could talk about that and why I sort of have that opinion, but I don't really think of them as fundamentally different. And I really don't think of them as different from things like exercise or different dietary modalities. In fact, I think of dietary modalities as just really, really dirty drugs in terms of biological effects. So all of these things are impacting biological systems, some of them with more or less specificity or with more potent specificity in terms of concentrations.
Incredibly potent.
Okay,
right?
I agree.
You're saying 5 or
but
10
dose
milligrams,
matters.
right?
Dose matters, absolutely. But fundamentally, they're all affecting biological systems in different ways, and so that's just the way I think about it.
Yeah.
Okay,
But
so,
like food is hundreds of grams, right? And rapamycin is 5 milligrams.
but there are people doing pretty extreme diets out there, right?
Well, to me, those critiques are kind of missing with me, because I don't have a 1 day a week faster, that I talk to. I don't have anyone in my life that fasts 1 day a week. And, extreme diets, I think that just doesn't work, so.
But are you as concerned about the 1 a day? Would you be psychologically as concerned about 1 day a week fasting? Okay.
Yeah. So,
okay.
So people that tell me that that go on really extreme diets like carnivore, or uncooked vegetables only, or 1 meal a day, I consider it sort of podcast disease. That they've heard it from an influencer, and it's generally not a sustainable practice, because people feel miserable. They have very low energy, and they're doing it because someone recommended it to them. And it usually just doesn't work. It doesn't just give people the sustenance that they need. If all of my scientific competitors wanted to go on 1 meal a day, my research group would win every time, because my people come in, they have coffee, they have breakfast, and they can go to work a day. I think that most people that are not eating until 4 o'clock in the afternoon are not going to have as much power as people that
Well, I'm definitely not a time restricted eater or a intermittent faster, but I do know people who it seems to work pretty well for them. So I guess I have a little bit of a different perspective. What about an aspirin a day? Why is that different?
Um, Largely, it doesn't work. We have a lot of clinical data on aspirin, right?
Mhm.
For a while,
But I mean, in terms of the way you think about it compared to like rapamycin once a week, you're more concerned about rapamycin once a week, I assume. Maybe not. Maybe I'm making an assumption there.
no, I think that I would say that for, aspirin and rapamycin, I would want to be guided by clinical data before making any
Okay.
recommendation to a person.
And I think that's fair. So the reason why I have sort of, and I think maybe, 10 years ago, if you'd asked me, I don't know if I would have been exactly where you're at, but I would have aligned more, I think, with the way that you think about these things. I think my view has shifted a bit, in part because I recognize that we're just not going to get that level of clinical data, right? This goes back to what I was alluding to before about how we would love to have randomized placebo-controlled clinical trial data for all this stuff. And I'm just realistic enough to know that's just not going to happen, at least in a time frame that I'm willing to wait for. So then I try to adopt my own sort of risk reward analysis for these things and choose. And I'm still honestly pretty conservative for this space. Like I don't take a whole stack of stuff. I'm pretty selective. I don't even take rapamycin all the time, but I've sort of settled on a risk reward analysis given my knowledge of the information that's out there. I get what you're saying though about how being worried that me saying that is going to influence other people to go out and start doing whatever, right?
Yeah.
I totally get that. On the other hand, if I don't share what I'm thinking and what I'm doing, you can ask the question like, is that the right thing to do? So I don't know the answer.
One thing that is interesting is that we have discovered things from athletes juicing.
Sure.
So,
Learned
for
a
example,
lot.
2006, Floyd Landis is way behind on the Tour de France, right? He's up in the mountains and he's experiencing hip pain, bone-on-bone hip pain, right?
Yeah.
And the next day attacks the course, makes up kilometers on the people that he's chasing. And then we discover later that he's taken a huge shot of testosterone.
Right.
Right? And at that time, in 2006, if you were to look in biochemistry or physiology textbooks, they would say it doesn't work that way. You would have to take androgenic steroids over weeks and months with resistance training in order to build skeletal muscle mass and performance in order for it to help you juicing.
Right.
That's not what he did. What he was aware of basically roid rage, right? He was aware that there's an immediate early effect of testosterone on the brain that would allow him to get on his bicycle and not feel the pain the following day, right? And so now we actually have a literature on the immediate early psychological effects of testosterone that was known in the bodybuilding community and the bicycling community that was not known to physiologists and biochemists.
Right.
So it is true that people have discovered things from their, N of one experiments and their, the gym or their, their group of scientific and medical colleagues. And so I think as a public health practice, it's a risky proposition. And there for sure there are influencers out there that are taking lots of things that make no sense whatsoever.
I agree. Yeah. I mean, I totally, yes, we won't name names, but yes. And I mean, I don't know if you're familiar with this, but I've talked about it a few times. Like the reason I first took rapamycin was because I was diagnosed with frozen shoulder, right? And I literally could not go across the street and throw a ball with my kid. Like I couldn't make this motion. I couldn't sleep at night. Finally got it diagnosed, and the doc was like, there's really nothing that I'm comfortable doing. So I want you to go back to physical therapy, and it might go away in a year. And I was like, I can't live like this for another year. And then I started thinking, okay, I know a drug that targets age-related inflammation really well. Like we give it to mice, and we see these inflammatory markers go down. I'm going to give it a try, and it worked. Now again, placebo effect possible.
Yeah.
but this was so severe.
Okay,
I don't
well,
think so.
I just got a cue to tell you about 7 randomized controlled trials showing anti-inflammatory effects of nicotinamide riboside.
Okay.
Okay, human beings, right?
Take it away.
So, so so NAD, what is NAD? NAD is the central catalyst of life. There's 4 NAD coenzymes, and basically they're like the wiring inside of cells. They carry high energy electrons. So high energy electrons are harvested off of our food. They're put onto NAD, forming NADH, and then NADH donates those electrons in order for us to make ATP. I mean, that's how fundamental this is. And then similarly, electrons get captured onto something called NADPH, and NADPH electrons are used to generate lipids, to generate nucleotides, to generate more cells, to repair things, to detoxify free radical species, right? And so I wasn't trying to do longevity research. I was basically minding my own business, trying to figure out how an enzyme worked. And an enzyme called glutamine-dependent NAD synthetase. And like I was working on that, and then I realized that there were some unsolved problems in NAD, right? That's how my group discovered the NR pathway, nicotinamide riboside being an unanticipated precursor of NAD. It's actually the biggest piece of NAD that can get into a cell, and then it gets 2 different enzymes come in, convert NR into NAD.
So before
right?
that, how is the biosynthesis thought to work?
So bacteria do it in two different ways. Bacteria can make NAD from either aspartic acid or tryptophan. So they take an amino acid, they do a whole bunch of different steps. It's called de novo synthesis. It's like baking a cake from scratch, where you have no ingredients that looks like a finished product of cake. That there's very simple ingredients, that a lot of enzymatic steps to go all the way from an amino acid to tryptophan. Then there's called salvage. And salvage is basically like a chop shop where, you bust your car, right? And then somebody comes and takes your car off of the highway, and then they remove the 4 tires on it because they're still good, and they remove the rear axle because it's still good, and they remove the radiator because it's still good. And then salvages, you could take those parts and you can build back together another car, right? So the salvageable precursors of NAD are nicotinamide and nicotinic acid. Those were discovered in 1938. And then basically my group found that there's actually a larger piece of NAD called NR that can be converted into NAD. So actually, you want to know what I really think about NR? NR is valuable garbage, because basically inside of cells, there's 4 NAD coenzymes, these NAD, NADH, NADP, NADPH. And then as cells bust open, or as we eat cellular stuff, then we're breaking down the NAD into these precursors, and the precursors can go back into cells and get phosphates and other stuff added to them and turn into the coenzymes of life. So it's the cycle of life, is that there's coenzymes, cells get broken down, and then get turned into precursors, and the precursors go in. So, but the interesting thing about NAD is that we know that our ability to make ATP declines in aging. And we know that our ability to make ATP is disturbed in many different diseases. It's attacked in inflammatory conditions. It's highly disturbed in Alzheimer's disease and Parkinson's disease. It's depressed in coronary artery, peripheral artery disease, and heart failure.
And now when you're talking about the ability to make ATP, you're talking primarily mitochondrial generation of ATP.
So the mitochondrial biology is severely disturbed.
Yeah.
In fact, people with mitochondrial disease are walking around not only with low muscle NAD, they're actually walking around with low blood NAD. They're the only people I've ever seen in my life that have low blood NAD. So I don't necessarily think that people need NAD tests and NAD kits, because you would know if you had a mitochondrial disease, right? So you don't actually need to pay me or somebody else to get an NAD kit. I mean, your NAD is probably about 22 micromolar, in your blood. It's a very abundant compound. If you take a supplement, it'll be higher. But
And just what's your take on, I mean, I know there is the perception that NAD levels generally decline with age. What's your take on that?
Yeah, so not as far as we can tell in the blood, but in a lot of tissues that are under metabolic stress.
And that's in mice?
Generally, the data are from mice, but we have data in humans in a number of different disease conditions. So, for example, it's hard to get, people will readily give you blood and urine, right?
Yeah.
And no one's going to give me brain, right? But we had something like 60 samples from the UK of people that had surgery for various conditions involving their liver, right? And a lot of them had alcoholic liver disease. And essentially all of the people with alcoholic liver disease had a depressed hepatic NAD system.
And is that total amount of NAD or NAD-NADH ratio or both? Or is that
a bunch of things are disturbed? We don't have to go into
just
all the inside
in
baseball,
general.
but it's down. NAD is down. The NAD plus to NADH ratio is less favorable for fuel oxidation. And basically what we found by discovering two things, by discovering the NR pathway and the genes that convert NR into NAD coenzymes, as well as developing a technology called quantitative targeted metabolomics, that there's many different diseases and conditions in which the NAD system comes under attack. So something that you and I will never experience postpartum, which every new mom experiences. Now, I'm not talking about postpartum depression, which might be 10 or 20% of moms. I'm talking about the fact of having just given birth to a baby is postpartum. If we are to extrapolate from mice and rats, postpartum is the mother of all metabolic stresses. That, how we think about the liver as the most generous organ, it's always doing what the brain or skeletal muscle or another organ system wants it to do. And how moms are the most generous people and the most generous animals. Well, a new mom's liver is the most selfless organ system that I've ever seen. So a new mom's liver is not only mobilizing protein, fat, and carbohydrate from her adipose and her liver to the mammary for a biosynthetic program to make milk, it's also distributing NAD precursors. So the mom's liver NAD system gets depressed. She's circulating a higher level of NAD metabolome. And in a mouse, the mom's mammary NAD system is increased by 20 to 30 fold in order to support this biosynthetic program. So when we realized that new mother mouse and new mother rats were under this metabolic stress, we wondered what NR would do for them. And then we were able to basically get their liver to have the level of NAD system of a naive female and basically boost their milk production. And then it turned out they spent more time nursing the babies, maybe just because they had more milk. I don't know that was a direct, psychological effect.
Sure.
But the development, the neurodevelopment of the offspring of NR supplemented mothers was demonstrably better. And it had a lasting effect on them. Like their ability to do adult hippocampal neurogenesis was better when those were adult animals. So, I mean, NR is an amazing compound.
So has that been tested in people? I mean, because it seems like
it's amazing. Yeah,
it's testable.
So I was invited up to Sacramento. UC Davis and a bunch of docs in Sacramento are really interested in lactation. And I believe that they registered 2 clinical trials to test NR in new nursing moms. One of them is in the context of preemies, because when a mom has a premature baby, the baby is premature and the mom is kind of premature as well in terms of her developmental cycle of when she's ready to produce a lot of milk. And so there's a couple of clinical opportunities to test that. But one of the things that we found is the NAD system gets disturbed by double-stranded RNA viruses. So famously, COVID-19 is a double-stranded RNA virus, right? And there's two kinds of immune systems in the human body. I don't know whether it's been discussed on your podcast, but there's the innate system and then there's the learned, acquired immune system, right? So the innate system is just that there's certain shapes of molecules that our cells know are bad news, right? Like our cells can detect bacterial endotoxin, because there's no molecule similar to bacterial endotoxin that is made in a mammalian body. We don't need to wait for an antibody against endotoxin because it just has some carbohydrates and lipids and weird things on it.
Anytime the immune system sees it, it's like, take action.
That's the innate immune system. Double-stranded RNA is just interpreted as there's a virus.
Bad news.
There's a virus here, right? So COVID-19, right, is a double-stranded RNA virus. And so it turns out COVID-19 infection transcriptionally activates at the gene level 5 different members of the PARP superfamily. These are NAD-consuming enzymes. And actually 5 different PARPs that almost nobody worked on. Like not PARP1 or PARP2 that ever role in DNA repair, but 5 different PARP enzymes that nobody really thought about get turned on in a COVID infection. We found that in human data, like from corpses, and we found that in model systems and in cell lines and stuff like that. So we found that COVID infection was attacking the NAD system, and then we found a function in the cellular response to COVID infection that required NAD, right? And so we showed in a JBC paper during COVID times that NR and nicotinamide as well were protective against COVID replication. And then there are other groups that have clinically translated that.
So when you say it was protective against COVID replication, what does that actually mean?
That means that the virus, replicates to a higher degree in a cell that has a lower NAD status. And that if you boost its NAD with NR, then even though it's churning NAD through these PARP enzymes, it can sustain enough NAD in order to have an antiviral response.
So there are cell culture-based experiments?
That was cell culture, and then it was mouse, and now it's human recovery in Sweden and Turkey, that human recovery is faster with NR and 3 other over-the-counter molecules in a so-called metabolic cocktail. Yeah, and now there's NR monotherapy that is being tested as well in RCTs, especially in long COVID recovery.
Interesting. Yeah, I think long COVID is obviously still a huge problem, and I mean it's interesting the parallels between rapamycin and NR, because I think COVID's another case where there's data to support the idea that rapamycin may have some positive benefits. And so the question, this started with inflammation, right? So I wonder if that's sort of the common denominator.
Yeah, so the common thing is that in these inflammatory conditions, the NAD system comes under attack.
Right.
So there's now 7 randomized controlled trials. The first one was El Hassan in 2019. I think that was in Cell Reports, and I'm on that paper, in which people taking NR had strong anti-inflammatory activity. So meaning that people are walking around with, as we age, with elevated levels of IL-6, IL-10, tumor necrosis factor. Somebody asked me about IL-11 for reasons that we could probably discuss. And we've never looked at IL-11. And so to my knowledge, I can't answer that question. But there's a whole bunch of inflammatory markers, right? And inflammation's trying to help us,
Mhm.
right? So just like in that double-stranded RNA example, where there's a cellular response and then there's a systemic response that's trying to kill a virus, inflammation is trying to find a foreign invader, right, and inactivate it.
Right.
So sometimes NADPH is used to like shoot electrons at bacteria and kill things. And so the NAD system can churn in those situations. And then interestingly enough, when you restore NAD status in a human being, you get these anti-inflammatory effects. So I think that's pretty exciting because arguably inflammaging and the kind of persistent chronic inflammation is a major component of a lot of diseases of decline, including neuroinflammation
Right.
and neurodegenerative processes.
Right.
So I would say the use case for NR is in resisting many conditions of metabolic stress.
So if I just want to make sure I have this correct though. So the way that you're thinking about it, at least for now, is that there is this increase in chronic inflammation, which is probably driven by multiple different signals. It could be DNA damage, double-stranded RNA viruses, bacterial antigens, whatever. And then that inflammatory response is dysregulating the NAD metabolome, depleting NAD, generally dysregulating the NAD metabolome, and that by supplementing with NR, potentially other NAD precursors, you can suppress that dysregulation of the NAD system.
Right. So arguably that's a very important, fundamental process involved in longevity science and geroscience, right? The reason why I don't say that NR is a longevity intervention is I don't know how to do that trial, right?
Right.
I easily
it doesn't
know,
seem to increase lifespan in mice. I mean, I think we have to be honest.
Yeah.
Right?
No, it doesn't. So again, it wouldn't have been my presumption that it does, but I mean, 50% of Black 6 mice die of cancer, right? And the intervention testing program is interesting because it has different mouse strains, but largely things that are going to be cancer protective. It'd be interesting to know more about rapamycin. Is it lower cancer incidence when
it seems to be delayed? So again, I'm not 100% confident in the data that's out there, but the data that's out there suggests that the tumor burden at time of death is comparable. It's just that they die later. So the interpretation would be you've delayed the typical age-related cancer progression.
Yeah. But I mean, what I'm happy about is that, I started out basically in yeast and enzymes, and I didn't even do the first mouse experiment of NR. That was done by Johan Aurich's group, right?
Yeah.
So Johan had been chasing SIRT1 and resveratrol and finding resveratrol didn't really work the way it was advertised. And, Johan at the time thought that, he needs to activate sirtuins. And I think that if he were to look at the literature today, he would say, well, I don't even want to, activate sirtuins. But he looked at our paper from 2007. 2004 was our discovery of the NR pathway. 2007 was us showing that it's boosting cellular NAD in yeast, and it's increasing replicative lifespan in yeast by activating SIR2. And we didn't say that, therefore, it's going to be a human longevity intervention. But Johan thought, well, that's great. I mean, Brenner has this compound that increases SIR2 activity, so I'm going to do a high-fat feeding model in Black 6 mice, male Black 6 mice, and that what he found was that NR was blunting the weight gain, right? But he didn't really figure out the mechanism.
And has that result held up? I don't know the answer, whether other people have reproduced that.
We did in our lab. We had a 2016 paper. I think his was like cell metabolism in 2012 or something. But we did a thing where we did high-fat feeding, and then we also added streptozotocin in order to. So streptozotocin basically kills beta cells.
Okay.
So now you can push a fat mouse over into a model of type 2 diabetes. We had NR was blunting weight gain, reducing fatty liver.
Do you have any ideas on the mechanism there? Why would it blunt weight gain on an obesogenic diet in mice?
So energy balance is really a key, right? So you're either having to increase energy expenditure, lower energy intake, or in some way deal with calories, right?
Hmm.
Otherwise, you're not going to be affecting, body weight. The most fundamental mechanism that we found is that in an overfed mouse, um, you get an attack on the hepatic NAD system. And it's actually not even centered on NAD plus. It's centered on NADPH, which is the controller of reactive oxygen
Right.
species. So by maintaining the levels of NADPH, the mice were able to control their systemic inflammation. So you see lower levels of 8-isoprostane, which is one of these markers of reactive oxygen, lipid
Right.
peroxidation, and then kind of all things work better, and then the mouse can maintain its skeletal muscle mass and its energy expenditure. Once you start, an overfed, mouse, gets an entirely different body composition.
Right.
You get inflammatory cells that are invading the liver, that are inflammatory cells that are invading the skeletal muscle mass. Skeletal muscle, isn't disposing of glucose as well any longer. So we think that fundamentally, again, the control of inflammatory processes, in part by supporting NADPH, is really a protective mechanism.
Right. Interesting. So I want to talk about yeast, but before we do, I want to go back to the human clinical trial. So in your view, what do you think is the most compelling human clinical trial data for NR at this point? And what are some of the clinical trials that either you know about or that you see coming down the
Yeah.
line that you're
Well,
enthusiastic
I think
about?
there's 9, that are really important. There's 7, because it's been repeated 7 times. Trials show that in human beings that NR lowers the inflammatory burden as measured by IL-6, IL-10, TNF alpha, and other things. That's been done by multiple different groups. It's been done here in Seattle at UW, in a population of people that are at risk for heart failure. It's been done in healthy older adults. It's been done in overweight adults. I mean, that's a really beautiful and important result. There's a medium-sized, I would say smallish trial that came out of Norway in Parkinson's, in which NR was improving cerebral blood flow
Mhm.
in Parkinson's. The government of Norway funded that trial. At the end of the trial, they wanted all participants to have NR because, I mean, the effect was substantive. And they've enrolled a much larger group of people with Parkinson's disease, and that's an ongoing trial. And then there was recently a collaboration, and I was not involved in the NADPARC thing in Norway, and I wasn't involved in 6 of the different anti-inflammatory trials. But I am, chief scientific advisor of ChromaDex, which developed the technology that came out of my lab. But in a new publication in Nature Communications, there was a group of very good clinical researchers from University of Florida and Northwestern University that looked at a population of classical aging condition, peripheral artery disease. Okay, and that these people are not aging super well. They have a low walking speed.
Right.
Their walking speed is actually going to be worse when you come back and measure it in 3 to 6 months. I don't remember how long the trial was, but the control group, the walking speed declined. And in the NR group, the walking speed increased, and the difference in the placebo-controlled difference was statistically significant. It was a pre-registered clinical endpoint, that's the gold
Yeah,
standard, right?
absolutely.
Because I don't know if people, even I as a biochemist didn't understand how important that was, probably 5 or 10 years ago. When you pre-register your endpoint, it means you can't cherry pick
Right.
from 20 or 100 other things that you exploratorily investigate post hoc, right? And if you're looking for, let's say, a p.05 statistical significance, ballpark, every 20 things that you measure, you
Right.
could
You're going to get
you
something.
could get
Yeah.
something, right,
Yeah.
that looks like it was significant. But they did a power calculation. They said their primary endpoint is going to be the placebo-controlled walking speed. They got a positive result. And one of the co-authors of the paper, you know, is on the, National Committee for Peripheral Artery Disease that comes up with, standard of care and standard of treatment. They already had designed their second trial, which is a much larger trial with even, more stringent stats. But that looked, really positive.
Good. I mean, and to me that makes sense, right? So one of the things I was going to do, not to intentionally try to be critical, but sort of as devil's advocate, is at least this is my reading of a lot of the NR trials that I'm familiar with, is they haven't typically hit their primary endpoint.
Yeah.
And again, most of these are underpowered. Like there's all sorts of reasons why a clinical trial doesn't hit its endpoint, right? And so it's like you're saying, so they fall back on things that are probably important and relevant, but not the primary endpoint.
Yeah,
right?
Very frustrating for me. There's a paper I'm on, Dollerup et al. There's multiple Dollerup et al.'s, but I think this was 2018. And Dollerup et al. Got really excited when we published our mouse thing with NR, because we have overfed mice, right? We push them into a model of type 2 diabetes, and we showed that their random blood glucose and their fasting blood glucose was put under control by dietary nicotinamide riboside. Okay. And we also showed that they had control of fatty liver. Amazingly, we showed that it controlled diabetic polyneuropathy. So, which is a really big problem, right? So this group, reading our paper as well as a competitor's paper on NMN, thought the mechanism must be insulin sensitization. Okay. I think it's getting the liver to work better, okay, rather than directly insulin sensitization. But they thought, oh, probably the primary endpoint should be weight loss and insulin sensitization. So they do, and nobody can afford a proper 24, 26-week trial with hundreds of people, right? Because these are academic researchers. They're getting a supplement from a company in California, right? That's a small company. So they do a 13-week trial. They use a nice high dose. They show that dose is safe. But with kind of wishful thinking, they specified their primary endpoint of weight loss and HOMA-IR, which means insulin sensitivity,
yeah.
right? So, and by the way, the trial was of 60 and 70-year-old obese Danish men. Okay. By the way, these men are physically inactive. They drink, they smoke. They haven't played hockey in 30, 40, 50 years, right? So they're not a super healthy population, right? There's no lifestyle intervention. There's no coaching. There's no dietary change. So would you like to hazard a guess on whether they met their primary endpoint?
Well, I'm guessing not, since you're going through this story.
Clinical NR didn't set the world on fire in 2018. No, they did not lose weight. They did not gain insulin sensitization, right? So then we're pouring over all of the other things that they measured, and there's a remarkable effect on fatty liver. Okay, so the percent hepatic lipids, this is absolute percent, like the percent of the volume or maybe area of a photograph of liver that is adipose, that is fat, was something like 21% for day 0 on NR, and it was improved by about 10%. So like cut in half, like 20 to 10-ish. Okay. And in fact, the effect size was, I believe the effect size was 10 to 1. And the effect in the placebo group was almost nothing. Okay. The problem is that there was so much variability in what the starting hepatic lipids were, that it was a p.13, which is actually fairly close
Sure.
to statistical significance, but it didn't meet statistical significance, right? So I believe that if we were to randomize people on hepatic lipids, like everybody has to be between 18 and 21, and then you go for the amount of trial that a hepatic lipids goes for, which is 24 weeks, that NR could have potentially been positive in that. But, I'm just stuck saying that, right?
Yeah. So, I mean, again, in your view, why is it that clinical trial hasn't been done? This is what keeps coming up. Like there are all these things where,
Money, right?
okay,
Because it's an over-the-counter, so it's an over-the-counter drug. So it's cheap in a drug world, it's too inexpensive for somebody to want to spend, tens of millions of dollars, right?
Right.
If it's an $84 million a year global product, that company is not going to spend 10 or 20 million dollars on a huge
and
drug,
again, I'm
right?
not picking on ChromaDex, but I mean but the reality is, right, that the incentive structure is just not there for a company like ChromaDex or any other company in this space to do the definitive double-blind, randomized, placebo-controlled, FDA level phase 3 clinical trial.
That said, I would argue that ChromaDex has really done things the right way, right? So this is I mean, it's the first vitamin discovered in, I don't know, 50 years or something, right? Came out of an academic lab. And initially, the IP was licensed by people that didn't develop it. One part of it was licensed by Sirtuins. Another part of it was licensed by Shackley. I thank my lucky stars every day that it wasn't developed by either of those companies, because I don't think that they would have handled it properly. After Sirtuins, decided that they were not interested in it, and Shackley decided that they weren't going to develop it, the technology, the IP went back to Dartmouth. And ChromaDex basically called them up, and ChromaDex licensed all of the academic IP in the space. They licensed a chemical synthesis from the late Anthony Sauvé at Cornell. They licensed my foundational IP at Dartmouth. They spent millions of dollars on manufacturing processes. They worked with a company called Grace to get a patent on a crystal form. They put a lot of money into safety and there is actually an FDA dossier for food and supplements, because FDA, the F in FDA stands for food. And so supplements, the over-the-counter products, do have to get a couple of designations.
Yeah.
One of them is called generally regarded as safe, which we got. And the second one is called new dietary ingredient, which ChromaDex got. And it basically shows data that the toxicology has been done in animals. And then here's a process for, human safety testing. And I was involved in the early, human safety testing of it. And so it was really a safety first company. And then it goes out and it's available as a supplement. It says it's not to treat a disease or condition. I certainly wouldn't go out there and say it's a longevity drug. I can't even say it's for any condition.
Yeah.
But now on the basis of, clinical trial results, we can say, actually, guess what? Inflammation is a condition, right? There's 7 clinical trials that do, and now there's much larger trials.
But again, those weren't the primary endpoints in any of those clinical trials, right? I mean, if you go back and look at the clinical trials, the primary endpoints, maybe with the exception of 1 or 2, weren't actually achieved. So again, I'm not suggesting that it doesn't have anti-inflammatory
So
effects.
Peripheral artery disein, it was absolutely the primary endpoint.
Right, and that was walk speed, right?
And then in COVID recovery and in some of these anti-inflammatory, trials, I believe it was the primary endpoint. And so, I mean, and that's being done by, we would also criticize, companies if all of the data comes from
I'm not
in-house,
criticizing
right?
ChromaDex at all.
Yeah.
I'm more commenting on just the misaligned incentive structures and the frustration I think we all feel that we don't get to the point where we have, really large clinical trials to test. And I would put rapamycin for different reasons in that bucket,
metformin,
right?
we could talk about metformin.
Yeah, right. So again, I think there would be a lot of value if there was some institutional, organizational way to get these high priority candidates rigorously tested and answer the question, damn it.
Right.
Right.
But I mean, considering the centrality of NAD, considering the centrality of inflammation and the potential to affect neuroinflammation and neurodegenerative conditions, as well as peripheral neurodegeneration, in which the NAD system is clearly under attack by the SARM1 system. I mean, I think it's a priority area. And, NIH and other, nonprofit funding organizations around the world have put some money into this. So I think it's a good thing. Plus, there's competing technologies for boosting NAD. There's NAMPT activators that are being developed. There's nicotinamide itself, which was active in glaucoma in a mouse model.
Right.
And there's an NMN company. And
let's talk about that for a minute. So first of all, one question is, in your view, I don't want to get into the weeds about the whole what's the name of the company with NMN? Metro, right? And how they're trying to get it taken off as a new drug as opposed to a over the counter. But I mean, from your perspective, as a biochemist or a biologist, is there any good rationale for NMN to be a pharmaceutical drug and other NAD precursors not be pharmaceutical drugs? Like, what's the biological argument?
Okay,
there?
Well, the biology is that NR is the largest piece of NAD that can get into cells, and we showed that the ability for NMN to elevate NAD depends on nicotinamide riboside kinase, right?
Mm-hmm, right.
So we talked about the importance of genetics, right? When you validate a target and you use genetics to figure out, is it working the way you're working? So we know that NMN depends on NR kinase to work, which means, and we also have time-dependent data using quantitative mass spec, which shows the phosphate coming off outside of cells, going into cells, the phosphate going on inside the cells. So we understand that, and so NMN doesn't have a unique.
So you don't believe that there's
action.
an NMN transporter?
No, there's not an NMN transporter, and there's SLC 12A8, I think is what it was named. That's a sodium transporter. It's recognized as a sodium transporter by people that work on sodium transport, the underlying data in the paper that nominated that as an NMN transporter had things like, here is the 0-minute time point, here's the 1-minute time point. You can't mix the reagents, that fast. They don't have an assay force that distinguishes between cell-associated NMN and intracellular NMN. They use the term immediately. Nothing in biology happens immediately. Everything is a time-dependent process,
sure.
Right? And so there's no real science behind NMN being one step later than NR. In fact, in neurodegeneration, accumulated NMN turns on a bad actor in neurodegeneration called SARM1. So if you lipidate NMN, there are chemical tricks that you can apply to NMN to get it to go into a cell, right? So you can do what's called masking the phosphate. If you mask the phosphate and you can push NMN into cells, it actually turns on SARM1 and gets SARM1 to turn NAD into cyclic ADP ribose, which activates calcium signaling and neurodegenerative processes.
Like off-target effects.
So that's why I know, I'm very confident that the novel compositions of matter that were developed by Metro aren't going to be developed as drugs. What they have is basically a crystal form of NMN, right? And I think that they're doing the right thing. Now, I don't agree. I don't think that they, as a company, have ever claimed that NMN goes through the transporter and is uniquely different than NR. I don't think that they've ever made a statement on that. I think that they are doing the right thing in doing randomized clinical trials with their crystalline preparation. The fact that they started doing that before somebody else started selling dietary NMN precludes those companies from selling dietary NMN. That's the FDA.
That's a bureaucratic rule, not
Yeah.
but there's no biological
No.
rationale for that.
Yeah, it's something called DSHEA. There's the DSHEA Act basically is trying to prevent us as consumers from getting failed drugs or from getting somebody to take a patented medicine that is being tested. Like, let's say that you had a rapamycin analog that a company just next door to us in here in Seattle is developing, right? And then they publish their
just
chemical
happens to be
structure.
a company next door to us that's
Right.
developing a
And
rapamycin.
somebody could say, oh yeah, I want to sell that as a supplement.
Yeah.
And could synthesize it.
Sure.
They can't according to the DSHEA law.
But in this case, this is a molecule that's present in all of our bodies and every eukaryotic cell and bacterial cell on the planet, right?
Those companies in principle could have gotten new dietary ingredient status for it, but they didn't until after Metro started testing it.
Right. So again, it's a bureaucratic, like who did what first. There's no good biological.
I don't blame Metro, in this
I get
situation.
it. From a business perspective, it makes sense for them to do it.
From a human health perspective, they're going by the FDA rules, and the FDA rules say that as they are testing this as a drug, it's not to be available to consumers. But the thing is, the companies that are still here's the important point. The companies that are still selling it don't actually care about human health. They only care about dollars, and those companies have not done the kind of safety testing that a company like ChromaDex has done, right? And there isn't NMN in the product either. So there's actually
toxin. What do you mean? There is not NMN in the product?
No. Andrea Meyer published a paper recently that 21 out of 23 of the samples that she was able to source in Singapore, and she sourced them from North America as well as the Far East, when tested, do not have NMN in the product.
Zero. There's not just contaminants. It's like the product is not NMN.
Many of them have no NMN.
All right, we'll have to take a look at that, and we'll link to that.
And then there's two different ways that the companies are making NMN. They're either making it through a fermentation process or through a chemical synthesis process. If you go through a chemical synthesis process, then you have a pretty high likelihood of having something like toluene left in the preparation. And if you go through a fermentation process, then you have a high likelihood of having something like endotoxin, left in it. And so these are companies that have not done the safety testing. Even NMN reagent available to people in laboratories is a very suspect.
Okay. So I want to talk that I think leads nicely into this study that I know you're aware of, where in mice treated with NMN, they found evidence for kidney pathology, specifically in aged animals. And the mechanism that was proposed there was high levels of 1-methyl nicotinamide and a couple of other NAD byproducts from liver metabolism.
Yeah, so there's a paper where there was like an association study that showed that there were people that had some excess cardiovascular events that had some elevated levels of some circulating NAD metabolites.
Yeah.
I think it might have been methyl 2-PY or N-methyl nicotinamide.
Right. That's fairly recent, right?
Yeah, that's relatively. So she may be proposing a similar mechanism. I don't think that's what's at work. You can actually feed mice N-methyl nicotinamide. It's not an NAD precursor. It's already been methylated. And I don't think that you would find renal toxicity. So I'm not reviewing her paper, but that's a suggestion of something that could be done. That's not been seen for NR. I mean, my concern is, can you test the reagent? Do you know what you're really working with? I mean, she told me that it's all the reagents used up. She thought that the reagent came from a reputable source.
Right. My understanding is that same batch was used in other experiments that showed positive benefits in some tissues and organs, and was from Shin Amai, who I'm sure you know who Shin is if you don't know him personally, but one of the sort of leading researchers in NMN.
Yeah.
But you're speculating that there's some sort of impurity in the the NMN, which is certainly possible. I guess the question I've got is, do you know if anybody has specifically looked? Because I think their data is intriguing to me, because they specifically see kidney pathology in the aged animals. So you don't see the kidney pathology in the young mice. And so it's at least plausible, right, that high levels of an NAD byproduct that we know is cleared by the kidney, could impact kidney function specifically in aged animals where there's damage to the kidney or other insults going on. So do you know if anybody has looked specifically at kidney
I know that kidney, I mean, there's groups at UT Southwestern and other places that have found in acute kidney injury.
Mm-hmm.
I think that AWRX has found this, as well as a UT Southwestern group has found it, that in acute kidney injury, there are disturbed levels of NAD metabolites. There's actually low NAD. And for that reason, there are ongoing clinical trials to see whether nicotinamide or NR can support healthy kidney function in
Right.
aging.
Because there is some data in kidney disease models as well for positive effects, for sure. Right.
So yeah, I mean, this has got to be sorted out.
Yeah. And then kind of the last thing I wanted to touch on is, how do you think about the various NAD precursors and their potential therapeutic spectrum, right? And so again, one of the things that I've talked about is the idea that there's at least some evidence that NR, NMN get broken down by the gut microbiome into niacin and nicotinamide, get taken up that way.
Necessarily we talked about the cycle of life earlier, right? I was looking around for an apple or a piece of liver, and all I see is a tree. But the cycle of life is that we're heterotrophs. We eat stuff. We don't photosynthesize, and we break down cellular stuff into NAD. The body plan of a mammal is such that our intestines do some of the work. And so our intestines are breaking down stuff like NAD. It gets broken down into NMN. NMN can get further digested to NR, and NR can absolutely be disassembled into nicotinamide and nicotinic acid in the gut.
Yeah.
In fact, depends upon a bacterial enzyme to go from nicotinamide to nicotinic acid.
Right.
But we go back to the genetic models to see, is NR available to skeletal muscle and to cardiac? That's shown already genetically. So in two different labs, in two different models. So one model was done in Joe Bauer's lab, where they knocked out the skeletal muscle NAMPT. So they knocked out the enzyme that skeletal muscle uses to convert nicotinamide into NMN, and then that goes subsequently into NAD. And that those mice had a skeletal muscle, contraction problem. They had a very low level of ATP production in their skeletal muscle, and that was cured by oral NR. So they actually delivered NR in the water bottle.
Mm-hmm.
And, I'm sure that if you wanted to measure, time-dependent destruction of NR in the water, and for sure time-dependent destruction of NR in the intestine, you could measure that, and the people have quantified that. But the fact that oral NR cured the skeletal muscle phenotype means that some of the NR was available in the skeletal muscle.
Sure, but again, I don't know if this is the case, but couldn't it be the case that that's because you're resynthesizing NR in the liver, and then it's available to the skeletal muscle? Like, again, it doesn't.
Okay,
I guess the question in my mind is, there's two questions.
So I have an experiment that in cardiac that addresses that. So I'm basically, again, I'm minding my own business in the late 2010s in my then office in Iowa City, and I get an email from Matthias Merixet in Paris, and he says, "What the heck is nicotinamide riboside kinase 2?" I said, "Oh, it's one of my, nicotinamide riboside kinase genes." He said, I knocked out my favorite transcription factor in the heart in a mouse. His favorite transcription factor is serum response factor. And I'm getting a dilated cardiomyopathy in this mouse that is progressing to full-blown heart failure. And before the disease even develops, when I take the hearts out in this, it's called SRF knockout model, SRF cardiac specific knockout, nicotinamide riboside kinase 2 is the most highly induced gene. I said, wow, that's so fascinating. I wonder if it's part of the pathology or if I wonder if it's part of the homeostatic response to pathology. In other words, is it high-level expression of nicotinamide riboside kinase 2 going to like kill this mouse's heart because it's missing a transcription factor and it has maybe that transcription factor is a repressor of NRK2? Or is there something that's being disturbed in the NAD system in that heart, and that the nicotinamide riboside kinase 2 gene is getting jacked up in order to try to repair the heart, right? So, so we, get permission. We ship him nicotinamide riboside, and they do, it's basically a placebo-controlled experiment in this SRF knockout model, and the SRF knockout mice have a terrible ejection fraction. Terrible fraction shortening basically means that they have dilated cardiomyopathy. They're going into heart failure. And that NR corrected 8 different things that they could measure. And so, and that's oral NR. And by the way, in the failing heart, not only was nicotinamide riboside kinase 2 going up as the most highly induced gene, NAMT expression was going down. When we gave them nicotinamide, it didn't restore the NAD in the heart, and it didn't improve their ejection fraction. So it has to be NR because this is a genetic model, right? In this genetic model, NAMT expression is going down, NRK2, nicotinamide riboside kinase pathway, is going up, and oral NR is available to that failing heart. And that's why your former colleagues at University of Washington are testing NR in heart failure.
Sure. That's interesting. And so I agree that would certainly be consistent with the idea that supplementing with nicotinamide in that model is not sufficient to raise circulating NR levels enough to offset the deficit.
Yeah. So glaucoma, I think it was Pete Williams got a positive result with nicotinamide, but there's just a brand new paper in the last 1 or 2 weeks where there was a group from Japan that used NR. I contacted them because I wanted to know whether they did a head-to-head, right?
Yeah.
And he said, based upon gene expression, we used NR because a lot of these systems, when there's some type of attack on the NAD system, the nicotinamide riboside 1 or 2 gene gets ramped up. And so that's part of the use case of NR is that it costs less ATP for a failing cell to make, so basically they have to make something called PRPP, which depends on glucose and a lot of ATP. So if they're making NAD from nicotinic acid, nicotinamide, it costs more ATP equivalents to restore the NAD system. In many of these systems, when COVID infection, nicotinamide riboside kinase genes are really upregulated. So it looks like the body is looking for NR. In my previous, nomenclature, it's looking for this valuable piece of garbage, this valuable salvageable precursor that it can turn, and it's a more valuable precursor than nicotinamide because it doesn't need that ribose unit.
Right. Now that makes sense from a metabolic perspective. So, I mean, I guess the question in my mind still is, I guess we don't know at this point, because there haven't been the sort of as many head-to-head studies as one might like, what the relative use cases are for niacin, nicotinamide riboside, nicotinamide mononucleotide. But it's your speculation that NR is probably going to have a broader use case because it is metabolically less costly to get to NR starting from
for sure. If it's addressing a condition, what I like to tell people, I've gotten calls from like rare disease foundations and all sorts of things that something about the NAD system is disturbed, and do I think that NR will work? And like, I don't consider it magic, I don't invoke, sirtuins. I don't invoke, you know, anti-aging expectations. But I tell people that if you see at the gene expression level evidence that the nicotinamide riboside kinase genes are upregulated and that the NAD system is under attack, then I think there's a very good chance that NR will be available to that tissue. And like, or if it's liver, then we know that NR is profoundly available to liver. It expands the NAD system in
Yeah.
liver and
Yeah.
in
Yeah.
a
Right.
unique time course with respect to nicotinamide and nicotinic acid. But in glaucoma, in retinopathies, in neurodegenerative conditions, in a lot of these conditions, the use cases are from gene expression studies that are showing that NR kinase genes are upregulated in these conditions.
Okay. And do you take NR?
So I don't tell other people, how much to take, but yes, I was the first person to take NR. And like, in terms of N of one things, like, I wasn't trying to, improve my athletic performance or anything, but I actually needed to develop the mass spec conditions and the blood extraction conditions so that I could get data
Yeah.
on an N of 8 or N of 12 clinical trial. And so, yeah, my blood was
You
drawn
sacrificed your body for
at
science, huh?
University of Iowa over a time course. So there's a paper, I guess it was 10, 10 plus years ago. It says a healthy 52-year-old male,
Uh-huh.
took a gram of NR. That was me at the time. I'm
That's
now
funny.
62. Yeah.
Yeah, awesome. Okay, so a couple of things that we haven't touched on yet. So that was great. Thank you. One, I think let's talk about sirtuins for a minute. So you've sort of a couple of times said that you're certainly not somebody who says that the reason NR has these effects is because of sirtuins. What is your sort of thoughts on sirtuins and their role in longevity
Yep.
and biology in general?
Well, I mean, you have some of the most important, citations in the field, right? So in the second half of the 90s, you and your colleagues in the Guarenti lab are showing that a bunch of genes that were earlier discovered by Jasper Rhine and Amar Klar involved in
Right.
mating type regulation, had a role in replicative lifespan, right? Which is basically the number of times that an isolated yeast mother cell can produce daughter cells, right? And then, you're in your own lab in Seattle, and you and Brian Kennedy and Stan Fields and others are saying, well, it's a little bit more complicated than that, right? And depends on strain background and a bunch of other things. And the simplified sirtuin hypothesis has kind of been overstated, right? And then when in the early 2000s it was claimed that 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 not only is the yeast SIR2 gene a longevity gene in yeast, but it's a longevity gene in worms and flies,
Yeah.
right? Which David Gems, in an accompanying editorial in Nature, when the worm paper came out, Tissenbaum and Guarenti paper came out, he said, this is kind of astonishing, right? Because it's as though yeast is like anticipating a cause of death or cause of aging in an animal, right? It's even more astonishing when you think that replicative lifespan, there's not a lot of mothers, right? So the ability of a yeast mother cell to divide a 21st or 22nd or 23rd time, there's like 1 out of 5 million of the cells that are that old, and you can regenerate the, right? So there's no selective pressure for that type of aging. So
hold on. Why is that different from any other type of aging?
Because Hamiltonian forces of natural selection. So if something has a conserved gene for aging, it means that offspring that have that form of the gene and that don't have it mutated will have the benefit of that. So yeast SIR2 is certainly conserved for gene expression,
Mhm.
right? And the ability for haploid cells to behave as haploids. But you can't say based upon the replicative aging model. I think there's a deeper critique of SIR2 than this, but
Yeah, I
based
agree.
upon
But I want to talk about this because this is something that I hear people get, at least in my opinion, wrong. So I want to try to understand. I want to try to make sure I understand your
if you
perspective on
can,
this.
my perspective is that if a phenotype emerges in only 1 out of 5 million cells,
Mhm.
it's not a selected phenotype.
I would agree with that.
Right.
But you said that's different from aging in any other organism, and that's where I'm asking, why is it different? Because I would also argue that lifespan is not selected for, at least not to any large extent,
I
in
would say
other
the
organisms
lifespan
as
is
well.
an emergent property, right,
Right.
of a very large number of genes that depends upon all of our organ systems functioning, right? Like if you're a squirrel or a bird or whatever, and you lose the ability of your liver or your heart, your circulatory system, or your immune system, or your brain to function, you're a dead squirrel or bird, right? So it involves thousands of genes and dozens of organ systems, and the longevity is an emergent property of many different genes and all of the organ systems working together that allow the animal to grow to be a replicating, sexually functioning individual, and in some cases, allow them to live another year in order to have another sexual
Right.
cycle
But that sounds like
and give off their
development,
genes.
not aging, right? So again, this is where I think, so here's the way I think about it. I think about the replicative aging model in yeast. And again, just for people who aren't in the
It's
weeds here,
very
right?
inside baseball.
So the way this is done is you have a single yeast mother cell, and it divides what's called asymmetrically. So you can tell the difference between the daughter cell and the mother cell. And if you count the number of daughter cells a single mother cell can produce, it's finite. It's around 20. Okay.
Mm-hmm.
That's the replicative lifespan. If you think about the force of natural selection in a dividing population, it's even more inside baseball because we could talk about non-dividing cells versus dividing cells. But if we only think about a dividing cell population, the force of natural selection is not going to be acting, as you've said, on that 21st, 22nd, 23rd daughter.
Right.
It's going to be acting on doubling time. How fast is the cell dividing? That's primarily,
But SIR2 doesn't have the phenotype.
Exactly.
Yeah.
Exactly. So, but I think if you look in other animals, natural selection is not acting on adult lifespan. It's acting on how quickly do you get to reproductive age, how many offspring do you have, and are you having offspring at the right time given the environment? So in my mind, fecundity in the yeast replicative aging system is really much more about how fast the cell is dividing, not how many times it divides.
Mm-hmm.
And I actually think that has a lot of parallels to aging or longevity in natural populations of animals, where natural selection is acting on rate of development, how quickly you get to reproductive age, and how many offspring you can have, again, within the constraints of the environment, making sure that you reproduce at the right time. There's almost no selection on adult longevity, especially in like natural populations under predation, right?
Right. So a fox would be a good example, right? So foxes in the northern hemisphere are born in the spring, right? And they are, pair up sometime over the course of that, summer and fall, and reproduce, right? And then the baby foxes are born in the following spring. So if the fox is not a very clever fox, or it's not good at avoiding predation, or it's not good at finding food, or it's not good at navigating, in the snow, then it may not replicate twice. It may not, right? But if it's a really clever fox and it's really has great sex appeal, then maybe it will live 6 years and it will have 6 times as many baby foxes, right? So it is putting into the gene pool successful foxes that's why I say it's an emergent property, because it's integrating its brain function, its muscular function, its navigation ability, its appearance, its ability to smell, and all the things, right?
Sure.
So I would argue that there's a bit of selection for longevity because the longer lived ones are going to have more partners and are going to have more
Yeah, I don't
offspring.
actually know that the data support that, especially if you take animals out of their natural environment and bring them into a safe environment, which we could argue is
Yep.
what has happened to humans, right? Certainly, if you think about mice in the wild, very few mice make it past one season, right? Because of
Yep.
predation, right?
Yep.
You bring them in the lab, suddenly they live 3 years in
Yep.
the laboratory environment. So I think we could debate about like
Yeah.
how much natural selection focuses
Yeah.
on longevity,
Okay.
but it's not a lot,
Yeah.
I would say.
Well, so anyway, let's.
I actually think
leave-
this is important though, because people will argue about the idea of, are there longevity genes? In fact, I think this is another topic I've heard you comment on, right? The idea that there are longevity genes that promote longevity, that function to promote longevity.
So we've talked about the monogenic longevity genes where loss of function extends lifespan.
Yep.
Right. Those are clearly could be called longevity genes.
Yep.
But from a biological point of view, they're kind of growth and development genes,
Yep.
right? Yeah. And then, but I would say that sure, there are longevity genes, but I would argue that something like human longevity or animal longevity, when it's not one of these extreme cases of monogenic loss of function, is hundreds or thousands of different genes, and that I don't expect there to be profoundly penetrant, high acting, alleles. I don't think that such
I agree,
things have
because they're
found.
probably selected against, because they have offsetting consequences that are not compatible
Yeah, I just
with
think that
the gene pool.
it's because, animal development is so complicated. And if you start saying that longevity is an emergent property that requires all of the different systems, then you wouldn't have as much of an expectation.
So let me ask you a question. So do you think that are you suggesting that longer lived species are fundamentally different in this way than things like mice? I'm not sure I follow. Or are you saying something else? Because we
No,
know,
I mean,
again, we know
there's-
in mice there are single gene mutations that can substantially increase lifespan in the laboratory.
That are gain of function.
FGF21 is one.
Mm-hmm.
So they do exist.
Mm-hmm.
So I guess I don't know the answer. Like
Mhm.
there are people out there who will argue that because humans are exceptionally long-lived for our body size and compared to
Mhm.
most other animals, it's going to be much harder to have big effects on lifespan in people. I
Mhm.
think that's a different argument than the question that we're talking about.
Mhm.
The other thing I would say about, again, we've sort of gone off on a tangent,
Yeah.
but this question of gain of function, I don't know the answer. You could be
Mhm.
right that that's going to be much more rare,
Yeah.
but I would also say people just haven't looked. So there have, all
Well,
of
people
the
are looking.
no,
right.
all of the large scale
Near
screens, in humans for genetic
Yeah.
variants, but
Yeah.
all of the large scale screens in model systems are loss of function screens
Yeah.
to look for genes that affect lifespan. So
Yeah.
the reality is we've just done a lot more looking,
Yeah.
at least in the model systems, for loss of function
Yeah.
than gain of
Yeah.
function.
Yeah.
But again, when you start talking about human naturally occurring variants, I think all bets are off, because my speculation is significant longevity variants are going to, in almost every case, have detrimental effects on fitness that are not going to be selected into the gene pool, right?
Yeah.
This goes back to what I was getting at before. So let me just try this out on you. So this is an idea that I've been playing around with. So would you agree that there is very little natural selection for lifespan in humans or in most other animals? There might be some, but it's not the predominant force
Right.
of selection.
I agree.
Okay. So it follows from that, I think, that at the individual gene level, few, if any, individual genes have been optimized for longevity in terms of natural selection, optimizing their expression level, pattern, whatever, for longevity. It's going to be optimized for mostly reproductive fitness
Right.
and
Right.
responding to environment.
Right.
So then it follows from that most genes you could, in fact, optimize for longevity. And you might get small effects in most cases. But if that's the case, then a really large number of genes might actually be longevity genes.
Yeah, I wouldn't argue against the idea that a large number of genes are longevity genes.
It might just be really, really small effect size.
That's what I think.
Yeah.
Yeah. But the amazing thing, it's interesting that you talk about FGF21 for an entirely different reason, we have a big project in the lab on
Really?
FGF21.
I
And
did not know that.
actually have a small molecule.
Well, let's talk about that.
Well,
I'm interested to the extent you can.
Yeah, I mean, we have a strategy. We understand quite a bit now about regulation of FGF21, and we have designed small molecules that increase a mouse's ability to make its own FGF21.
Oh, interesting. Yeah. So I don't know how familiar you are with the FGF21 literature. I'm sort of familiar with it, but I interviewed Crystal Hill, who studies protein restriction. And so she was taking me through the protein restriction literature, where if you protein restrict, you increase FGF21. That's required for the lifespan extension.
Yeah. And
then there's transgenic FGF21. They express FGF21 at a much higher level than you get from protein restriction, but it's a pretty big effect. It's like, 30+% effect on lifespan. I don't know that that's been independently reproduced yet, but it kind of lines up. The thing that I find intriguing about FGF21, and I might get the biology wrong here, but this is the way I understand it right now, is that FGF21 interacts with, I think it's alpha klotho, right?
Beta klotho.
Is it beta klotho? Yeah. Okay, so then it's alpha klotho that is in the brain primarily. There's two of them, right?
Beta klotho is the co-receptor with the FGF21 receptor, receptor, and it's beta klotho in the hypothalamus that mediates the aversive response to sweets. And then there's beta klotho somewhere in like the rear brain or something that is aversive to ethanol. But it's really interesting system, right?
Right. But both of the klothos have been implicated in longevity now, right? Which I find intriguing, given that people don't really talk about them in the same breath in terms of the longevity field. But to me, that's like, come on, there's got to be some overlap there. Yeah. So it's super, super interesting and intriguing. So anyways, that's cool that you're working on FGF21. And I think there's a lot of interesting biology there as well. Does that tie into NAD, or was that completely separate?
It does sort of tie in with NAD. Basically, I got an inquiry from a rare disease foundation that disturbs one aspect of the NAD system.
Interesting.
And then we found downstream of that some disturbances in FGF21 regulation, and so that's turned into a huge project in my lab.
Okay, so that was sort of a tangent, but we started on sirtuins. So let's come back to sirtuins and where we left off.
Well, again, I was giving you credit for multiple contributions to sirtu biology, and in 2011, in Burnett et al. in Nature, with Linda Partridge, David Gems, and some others. Re-examination of the original, worm result and the original fly result showed that no overexpression of, the SIR 2.1 as it's called in C. elegans, or SIRT1, DSIRT1 in flies does not extend worm or fly lifespan. So basically, the whole thing got overblown. As though, some people to this day claim that they identify dominantly acting longevity genes in yeast and that they extend lifespan in worms and flies, and they don't. And that they extend lifespan in mice, and they don't.
Yeah. So I everything you said is true. I mean, at least in terms of the chronology of the publications. A couple things that I would say. I don't take quite as harsh a view of the sirtuin literature. So from my perspective, the original study in yeast, you overexpress SIR2. That clearly works. Does it work in every strain background? Probably not. Does it work in most strain backgrounds? As far as I can tell, yes, but I don't know that anybody's looked extensively. Okay. But that I think everybody agrees has stood the test of time. The C. elegans and fly literature is tough. Like, I actually don't know where the reality lands. And as you can probably appreciate, right, when you get differences of results, or irreproducible results, sometimes it's very difficult to know why. Sometimes it's clear. Like, I think the resveratrol case is clear. We don't have to go into that, but I think it's clear. In the case of the overexpression of sirtuins, it's super complicated because of the transgene used, the dose used, the conditions of the experiments. I actually don't know. I don't know what the real answer is. It wouldn't shock me if there are certain doses or conditions where you can get a smallish effect on lifespan from overexpressing worm or fly SIR2. I think the mouse situation is interesting in two ways. There are 7 sirtuins in mammals. People have tried really, really, really hard to find
That's
lifespan
like being
extension.
followed by 7 police teams, right?
Right.
You're going to observe something.
Yes.
Right?
And people have tried really, really hard. And like you're getting at, yeah, if you try an experiment enough times, you're going to get the result you want eventually. So that's the thing that makes it hard to give credibility to the idea that sirtuins promote longevity in mammals. Having said that, there are 2, studies that on the surface, if we didn't have all of that other stuff with SIRT6, that actually look pretty good. And so I don't know, again, could it be the case that SIRT6, because you can make you can come up with a plausible biological rationale that SIRT6 is probably functionally the closest to yeast SIR2 in terms of what it does, in the sense that it definitely regulates silencing and genomic stability in mammals. And that's clearly what SIR2 does in yeast, no question about it. So is it possible that SIRT6 is the magic SIRT, right, for mammalian longevity? I get it. No, I get it. No, I get why you're skeptical. I'm also skeptical, but I also think those papers they look pretty good. And so trying to balance, maybe I'm going too far and trying not to be overly cynical because of my history with sirtuins. I don't know. But that's sort of where I land. Like, I'm just kind of waiting for the dust to settle so we can figure out what's real and what's not real.
One of the problems is that we know that SIRT6 is a positive regulator of gluconeogenesis.
Mm-hmm.
Right?
Yeah, right.
That's clear. And we also know that mice are really different from humans in terms of the age-dependent glycemic regulation, right? So as humans age, our blood glucose tends to go up. As a mouse ages, its blood glucose tends to go down. So extra copies of SIRT6 help a mouse maintain its blood glucose when it's in its dotage, which is going to be really bad for humans.
Yeah.
So I think that the idea of a SIRT6 activator being useful to humans based upon what we know about SIRT6 being a positive regulator of gluconeogenesis makes it extremely unlikely that you're going to get SIRT6 medicine.
Yeah. Having said that, there are genetic variants that people have associated, with centenarians and extreme longevity in humans.
They have lower deacetylase activity.
Right.
And so they don't phenocopy the effect of higher NAD. It works the other way around. I have something in preparation right now, or might even be on ResearchGate right now, where I reviewed that literature. And if you dig into it, the molecular characterization of the SIRT6 so-called centennial allele doesn't behave in a way that would phenocopy a higher NAD status. It would be more resistant to a higher NAD status.
Interesting.
So I think it.
Yeah, I'm not. I haven't done a deep dive in that literature. My recollection was that the thought was that it had better DNA repair. Those alleles have better DNA repair activity. But again, it's one of these things where obviously when people design these experiments, they go looking for a certain outcome. And so you have to kind of weigh, how much did that influence the outcome of the experiments. But I'm still on the fence when it comes to sirtuins. But I do think, I guess where I fall is if sirtuins were really, central regulators of longevity, it wouldn't be this hard to find out whether or not they have a big effect on longevity.
I mean, there are, in my review, sirtuins are not conserved longevity genes. I cited 5 papers where they overexpressed SIRT1 transgenes. 4 were whole body, and none of those extended lifespan. And one of them was something about anticipatory feeding, and that didn't hold up. And then there was one where it was supposed to express in the brain, and they actually described 2 different lines that came out of that.
Yeah.
One they called line 1 and one they called line 10. I don't know what happened to lines 2 through 9, but literally line 1 did not extend lifespan, and line 10 did. And so you would after 4, whole body, experiments, and then 2 different lines in the lab, one which doesn't extend lifespan, most people would say it doesn't extend lifespan. There's a position effect in this particular transgene 10. But that's not what they did.
Right.
They came up with a post hoc explanation, and they said, oh, it has to be at this particular ratio. And that's not the way genes work, right? If something were selected for a longevity phenotype, then it would be a penetrant phenotype, right? And, it would be, if it were selected, you'd be able to put it into many different gene, strains. Just like when you knock out growth hormone, you get the effect in any gene, in any mouse strain that you want, right?
Sure.
It's a fundamental process. And that's where the sirtuin literature fails.
Yeah. Yeah. Again, I think, we'll see how it settles out, but I don't think there's a lot of reason to believe that sirtuins are potent regulators of
But
longevity.
the thing is that NAD is fundamental to life, right?
Sure.
And so we really like to focus on conditions of metabolic stress. Time zone disruption is one. Lactation, as we discussed, inflammation, noise-induced hearing loss. Sun exposure where you have DNA damage, PARP1 gets activated. There's a lot of these conditions of metabolic stress that disturb the NAD system. And that's nice because it gives us nice actionable endpoints in animal experiments and in human experiments where we can look for effects on NR. So I don't have to live 200 years to do a 90-year experiment on human lifespan.
Right. Which is, that's a good strategy to take. So given all of the importance of NAD and all of the places where NAD dysregulation could play a role, what is your sort of optimistic, well, two questions. Like one is, there are a lot of people taking NR now. What does that data look like? I mean, would you expect to see big effects in people taking NR in general, or is it going to be just a subset of the population? And what do we know? I mean, I know we've got these clinical trials that we've already sort of talked about, but in the much larger population of people who are taking NR supplements, what, if anything,
there's kind
do we
of
know?
a performance group of people, and there's kind of an aging group of people. And so, for example, Rudy Tanzi and I both advise ChromaDex, and Rudy is, during the Bill Belichick era, was literally always on the sidelines of the New England Patriots, and I was photographed, with Belichick as well. So it's widely understood that the Patriots were on Niacin. And so those are young people, well, except Tom Brady was 40, but those are generally young people that get a bruising on Sunday, right, and have to practice again on Tuesday, and then are playing again on the next weekend. And so there's people that are kind of take NR in order to boost their repair capacity, which I think is a really important theme. And then there's healthy aging, whatever that means. And so I'm excited for clinical trials. I actually promised on Twitter an out of the box idea for human aging model system.
All right, let's hear it.
So even though you and I both came up through yeast, right? And so mad respect for the awesome power of yeast genetics, and mad respect for worms, which are right next door to you, and flies. And we talked about mice, but mice don't die of falls, right? And they have a short lifespan. So what about the human condition, the human aging condition? Well, there's monogenic progeric conditions, right?
Yep.
But again, that's a very, very peculiar type of aging. A particular helicase is, disturbed. And so you do a study on lamin A mutations. Maybe you learn a whole lot about lamin A, mutations, and maybe not so much about general aging. But when we were walking through the biotech next door, somebody said something about astronauts, right? So they have, they're exposed to stuff out in space. They have, irradiation, and they experience some aging. But that's a very small population. So I'm at a cancer center. I'm at City of Hope Comprehensive Cancer Center, and we have a registry of pediatric cancer survivors.
Right.
I think this is a population where the genetics is just like the rest of us. I mean, maybe there's a slightly higher chance that they're BRCA1 carriers
Sure.
or
Yeah.
APC min, carriers or something like that. But largely cancer is an environmental disease, right? Largely they have the same genetics as the rest of us, but due to their treatment exposure, whether it's immune therapy or conventional cancer chemotherapy or targeted chemotherapy, all of these things have an effect of accelerating aging.
Yep.
So two of the people that were involved in my recruitment, Saro Armenian and Rusha Bandari, basically invited me into a super group of people that study cancer survivors. And we're doing some of our first interventions now, lifestyle being really key,
Yeah,
right?
of course.
So because, we want the standard of care to be physical activity. And then we want to see a marginal benefit of something like nicotinamide riboside. And so I think that that's a population where you can measure people's cardiovascular health and you can measure resistance to a decline, because just like the peripheral artery disease, these people are in a kind of premature decline of their cardiovascular fitness.
Is that sort of independent of the treatment modality that they got? I'm wondering how much of this is due to the type of treatment, the chemotherapy or radiation.
The great thing about the data and the population registry that they have at City of Hope is that experiment can be done, and that they've looked at.
But I mean,
outcomes
the accelerated aging phenotype, is that seem to be across the board, or is it dependent on?
It's relatively broad, and it's better characterized for some chemotherapies than others.
Yeah, interesting. Yeah, no, I mean, I think that makes perfect sense, and I think there are other sort of subpopulations that may also have that kind of accelerated aging. Or I think it's an interesting question. Is it a global acceleration of aging, or is it more like the segmental progerias and it's only in certain tissues and organs? Unknown at this point.
Relatively global.
Yeah, I think you're probably right in that case.
Right.
I think the progeria mutations are not. I think they are in fact segmental.
Yeah. Right.
But in cases like that of environmental, we're going to call it environmental exposures. I think it probably is pretty global.
So put that.
Yeah, interesting
in
to wonder too about how much of that is epigenetic, right? And so first of all, you could measure that, and people have done this, I'm sure, using the epigenetic tools that are out there for looking at at least epigenetic aging. Actually, that's another topic we can touch on before we finish up, because I'm sure you have some interesting opinions on the biological aging clocks. But I think this idea makes a ton of sense. The other population that I've thought about in this context is Down syndrome. So kids with Down syndrome, as they go into adulthood, show early onset for a bunch of different age-related diseases. And I don't know what the mechanisms are there. I mean, you know what the mechanism of Down syndrome is, but I think that's another population where they're aging rapidly, have early risk for a bunch of different age-related functional, not just diseases of aging, but functional declines, and potentially could have outsized impacts for things like lifestyle interventions or other interventions that might have an impact. So yeah, that's intriguing. Okay, let's finish up with biological age clocks. What do you think about that?
One of the problems is cherry picking, right? So there's a bunch of interventional studies where they used 8, so-called age test kits, and then they chose the one that they liked.
Sure.
And so that's not good. I don't think that the age tests provide, independent evidence that someone is doing better, or they certainly don't allow a person to say, I reversed my age.
Yeah.
Because you get a lower number on a kit. You can also kind of game them, right? So if you take, testosterone or growth hormone, there's going to be some physiological consequences for that. You're going to be circulating biomarkers of a person that has a, higher testosterone growth hormone status, whether that is going to be accelerating your aging or retarding your aging.
Yeah, I actually don't know the answer. I'm sure the answer is out there. True Diagnostic probably knows, but it's actually interesting. Would hormone replacement therapy affect, how does it affect epigenetic age? I suspect
many
trying to think if I've
many of
seen
those
data on
tests
that.
many of those tests responds to
Oh,
those
sure.
types of
It's going to
interventions,
respond. Yeah.
and I don't think that that's interesting. Like somebody said, oh, smoking doesn't accelerate age, and I said, what do you mean? We know smoking makes you age worse.
Yeah.
But
So let me ask you, why do you think it's not interesting? So again, we're using the phrase biological age test a little carelessly, but if we think about just the epigenetic tests that have been, highly correlated with disease outcomes and mortality, right? Because you can build the chronological age correlation, and then you can do additional steps to identify different subsets of marks that are correlated with either specific disease risk
yeah,
or
to some
mortality
degree, yeah.
risk. Yeah. So why do you think that it's uninteresting if an intervention moves that needle because you don't really you believe that it's just correlation and not causation, or do you think that we can't make any claims about aging from that?
I think it's undetermined in terms of what is these are biomarkers,
Sure.
right?
Of something.
And
Yes.
I don't think that they provide independent evidence. You have to look for functional performance.
I agree.
That's why walking speed or physical performance or grip strength or lung capacity, anything like that would be much more important to me.
Yeah, I totally agree with that. I mean, I think functional measures are absolutely the gold standard we
Yeah.
should be looking at.
Okay.
But I do wonder what these clocks are actually informing us on, and it's speculative, but it wouldn't surprise me if at least the current crop of epigenetic clocks is really a signature of health status, for lack of a better way of saying it. Like, your health status to some extent determines how likely you are over the next period of time to get a disease or die, right?
Yeah, but I don't think it has a value proposition to the consumer right now.
I agree with that.
It's a research
completely.
tool, right?
Yeah. No,
Yeah.
completely. So I'm thinking
Yeah.
about this more from a biological
Yeah.
perspective. That's why I always say these things are for entertainment value only, right? I think if you want to take the test and whatever you want to do with that, great. But I certainly would not. I would favor being able to lift something heavy or run fast or walk fast or whatever over a biological age
Yeah.
score, for sure.
Body composition, on the
Yeah.
other hand, like when I joined a gym, right, recently, and I changed my gym, and they gave me a body comp test.
Yeah.
And I thought that was interesting
Yeah.
because it was like the percent skeletal muscle mass was good, and it was like, but it's better in Brenner, it's better in your butt and your legs than it is above here. So if you're trying to figure out what to do more of this, or
Yeah.
realize that you're way ahead of the game in terms of your low rise. So I thought
Yeah.
that was useful. I
Yeah.
mean, that was
We use
something I could use
DEXA
that.
all
Yeah.
the time, and I think that's one of the more useful diagnostics. I assume that's what you got for body comp, unless
Yeah,
it was
I think
like
it was DEXA, yeah.
it was where you lay down and they take a scan of you.
No, I didn't have to lay down for it, so I don't even know what it was. but-
Okay, yeah.
yeah.
So did you get bone density? Because that's another
No.
thing DEXA will give you.
Yeah.
Yeah. So that's actually something that's important to pay
Yeah.
attention to. Although in my case, I have really, really high bone density. I'm like 3 standard deviations above the mean. It's all in my head. It's like, if you look at the bones in the rest of my body, I'm a little bit above average, but my head is like that thick. So
Thick, huh?
Yeah.
Wow.
proof positive that I have a thick skull. And with that, we'll finish. No,
All right.
anyways, I think this has been great. We've covered a lot of ground. Anything we didn't talk about that you want to talk about?
Well, I just like to emphasize that in terms of my my both intellectual interest and financial interest in NAD, I think that my track record kind of speaks for itself, like showing that NR, increases lifespan in yeast, and then being the one person in the field saying, no, that's not going to make it into a human longevity medicine, despite the fact that if I were to jump on board the sirtuin train, or just saying that SIR2 mediates the beneficial effects of NR and NAD, blah, blah, blah. So, I pride myself at, evaluating data. I had a meeting just a couple of weeks ago with a group of people that wants to test a NAMT activator in idiopathic lung fibrosis, right? And that could be considered a competing technology. But we want to understand what is the best way to elevate NAD in various different tissues. It's going to probably depend upon various different technologies, formulations, and preparations. It's going to require people to pre-register their endpoints, leave their egos aside, and not exaggerate the data. So that's just what I try to do.
Great. I appreciate that. And I think, yeah, I mean, people are going to naturally gravitate towards, conflicts of interest and focus on that. But I think, in my opinion, you've always been very transparent. And, I think even though we've had our disagreements, certainly publicly on Twitter, as I've said before, respect your rigor and your approach to science. And I think you've handled the conflict of interest, about as well as you possibly can.
Thanks. I appreciate it.
Anyways, appreciate you again coming up here
Pleasure.
for the day. It's been a lot of fun, and I look forward to the next time we can sit down and chat.
Okay, sounds good.
Thank you. All right. Yep. Thanks everyone for tuning in to the Optispan podcast. As always, if you have any questions or comments, please put them in the comment section below. And if you haven't hit the subscribe button yet, hit the subscribe button. And I hope to see you next time on the Optispan podcast.